Hello everyone, and welcome to today's live broadcast, Isolation and Characterization of Exosomes and Ectosomes. I am Dr. Chad Schwartz, Senior Application Scientist at Beckman Coulter, and I'll be your moderator for today's event. We are delighted to bring you this educational webinar presented by LabRoots and sponsored by Beckman Coulter. Beckman Coulter is a leader in centrifugation and flow cytometry and has long been an innovator in particle characterization, lab automation, and genomics. Beckman Coulter products are used at the forefront of important areas of investigation and discovery. For more information, please visit beckmancoulter.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing this presentation, please click on the support button at the top right of your presentation window or submit your problems through the green Q&A button. I would now like to introduce today's speaker, Suresh Mathivanan. Dr. Mathivanan is a laboratory head in the Department of Biochemistry and Genetics at La Trobe Institute for Molecular Science. The Mathivanan Laboratory developed and maintains two widely used databases for exosome and extracellular vesicle research, namely Exocarta and Vesiclepedia, and has been performing pioneering research on exosomes for the last 8 years. Today he will tell us about a global proteogenomics analysis that his lab performed, which has unveiled the secretion of mutant proteins that are implicated in cancer progression through EVs. Before we get started, I would like to ask a few poll questions. Our first question is, what separation technique are you currently using to isolate exosomes? Ultracentrifugation or differential gradient, affinity capture, size exclusion, or precipitation reagents? We'll give the audience 15 to 20 seconds to answer, and I will come back with the results. All right, great. The majority of folks use ultracentrifugation or differential gradient at 62.3%. 13.2% of you use affinity capture. Size exclusion comes in at 6.6%, and precipitation reagents at 17.9%. Our second and final poll question is populating now, and it is, what is your sample source for extracting exosomes? Either tissue, cell culture, blood, urine, plasma, or other. Again, we'll give the audience a bit of time to answer. All right. It looks like the majority of everyone in attendance here is using cell culture at 41.9%. Plasma is our second highest at 20%, followed by blood, and then tissue, and then urine, and there's also an other at 10.5%. Thank you, everyone, for your answers, and I will now turn it over to Dr. Mathivanan for his presentation.
Thanks, Chad. Thanks for the kind introduction. So today, as we spoke about, I'm going to talk about isolation and characterization of exosomes and ectosomes. These are two different types of extracellular vesicles that the cells secrete in physiological and pathological conditions. So I'm just going to share our lab's experience in isolating and characterizing these extracellular vesicles. So prior to going into the extracellular vesicles, we all know about the classical and non-classical way of protein secretion. Most importantly, the classical secretion pathway, which involves the ER and Golgi. This has been very well characterized over the years. And the protein which is about to be secreted should have a signal peptide, and then that gets cleaved off, and then these proteins are then targeted towards to be secreted out from these cells. So recently, or for the last 3 decades, we know about some of the ways that the non-classical secretion mechanisms, or by which proteins are secreted other than the ER and Golgi pathway. For example, like we know about exosomes, which are small vesicles that are secreted out by the cells, and these doesn't follow the canonical or the classical secretory pathway. Similarly, we have something called as ectodomain shedding, or shedding of plasma membrane. So imagine there are a lot of plasma membrane proteins which are present in the cell, and these can be cleaved off by extracellular proteases, which can then cleave these particular domain, or which can be the ectodomain, and that can be shed into the extracellular space, and that can have a functional implication on either stimulating the recipient cell, or then trying to do some signaling pathway in the recipient cell as well. And in other ways where there are other mechanisms by which a protein can pass through the plasma membrane channels through there, and this is a rare mechanism where not many proteins are being reported by that kind of mechanism. And lastly, I just want to emphasize on the shedding microvesicles, or alternatively, which are called as ectosomes, which are coming from the plasma membrane blubbing. Again, there's a controversy in the field where we talk about these vesicles as together, and we sometimes call them in a mixed way where we call microvesicles for both of them. And there's no standardization as such, but people in the field are trying to create one. And currently, this is what we have. So let's focus on exosomes. These are secreted nanovesicles from the cells. As you can see, once there's a receptor which is internalized, and then that leads to the formation of early endosomes. The early endosomes then mature to form late endosomes, where there are intraluminal vesicles which are happening by the invagination of the limiting membrane. And this results in the formation of small intraluminal vesicles inside the late endosomes. So now the late endosomes can partake 2 functions. It can either go into the degradative pathway where it can fuse with the lysosome, or it can take the exocytic pathway where the late endosomes can fuse with the plasma membrane and release its contents into the extracellular space. So upon release, the intraluminal vesicles, which are the small vesicles which are present inside the late endosomes, are referred to as exosomes. So recently, a lot of studies have shown that these exosomes have a lot of physiological functions where they can stimulate signaling pathway in the recipient cells, as well as transfer the proteins and RNA content to the recipient cell. So what we have done in the lab, like we have isolated an exosomes from cell lines and blood plasma, which is good that a lot of people in the poll that Chad conducted had people have been isolating exosomes from cell culture media and also from plasma. So we had done a comparative analysis to see which methods are better in isolating exosomes to homogeneity. When I say homogeneity, again, when we look in terms of the vesicle characteristics, we also look in terms of markers which are known to be present in the vesicles, the extracellular vesicles. And then we compare these 3 methods and say, okay, which is the method that which is not as cumbersome, but also isolates a purer population of exosomes. So we use the commonly used methodologies that was currently at that time. So this was probably now 3, 4, 5 years old. Especially the cell line study was done 4 years old, but the plasma study was done 2 years ago, which was published in 2013. So we initially took differential centrifugation coupled with ultracentrifugation, which is currently the gold standard in terms of isolating these vesicles. When I say gold standard, because all the other techniques we have, even though they may be robust, there are some inherent problems with some of the isolation methods. But if you want to capture the whole population of exosomes, currently a lot of the labs use differential centrifugation and ultracentrifugation, and is one of the instruments that is available to many labs, and where we not have to have any specialized instruments for these. So this is what we did. We isolated exosomes from differential centrifugation. Then we also did an OptiPrep density gradient centrifugation. Previously, a lot of researchers used to use sucrose gradient density gradient centrifugation as well. But based on prior literature, a lot of people were able to show that the sucrose density gradient centrifugation was not able to separate viruses from extracellular vesicles, as well as sucrose is not, it's toxic to the cells. When you take the extracellular vesicles from these and try to do some functional study with that, you might introduce some toxic biases to the culture media. And also the OptiPrep density gradient solution have an isoosmotic solution, which keeps in terms of there's not much osmosis in there, and you might not have sample loss, and your exosome integrity is preserved. And most importantly, when you're trying to do some functional analysis, the exosomes are indeed functional for quite some time. So lastly, we used immunoaffinity pull-down approach, which people used, and in this case, we used EpCAM, but this can be used on any other surface antigen that we think is present in exosomes. The main reason we started using EpCAM was these kind of beads were commercially available, and it is easy for many labs to purchase and do the analysis. Plus, prior to that, 2 other studies isolated exosomes from serum of ovarian and lung cancer patients, and they were using EpCAM antibody beads. So the purpose is to identify which are the better methods in isolating exosomes, and then we compare these methods and say what is isolating purer population and what is easier to use, and things like that. So this is a summary of the outcome, which was identified from the 2 studies. As you can see, when we use the cell line culture media, again, this is, when I talk about the cell line, it is a colorectal cancer cell line called LIM1863, where these are organoids. These are not adherent cells. These grow as suspension. And these cells were easy to culture, and we use that for all our purposes. So when we did the differential centrifugation from these, and when I say differential centrifugation, it is isolating the exosomes by 100,000 G. You spin them down for 100,000 G for 1 hour, and you wash them with PBS, ice cold PBS, and then you have the pellet, and the pellets, we call it as crude exosomes. And OptiPrep density gradient centrifugation performed better than the density gradient centrifugation, as you can see here. But the important thing was the immunoaffinity pull down performed better than the OptiPrep and density gradient centrifugation here. But as the immunoaffinity pull down was done on EpCAM, which is epithelial cell adhesion molecule, which is present in a lot of epithelial cells, we were able to isolate them. And when we looked at the proteomic profile of the exosomes isolated from these 3 methodologies, we were able to get more exosome-like signature in the immunoaffinity pull-down. I have to acknowledge is that this particular study was done in, when I was a postdoc in Richard Simpson's lab, who is well known for his proteomics work and isolation technologies. So later on, when we started the group, we also were interested in plasma, blood plasma, because we wanted to do a lot of patient samples, and we wanted to identify biomarkers and validate those biomarkers in the patient samples in a robust and easier way. So for that reason, we compared these three methods in terms of the normally used methods, which is the differential centrifugation, OptiPrep, and immunoaffinity pull-down. So one of the problems with plasma, as we all know, in terms is it has a lot of proteins which are highly abundant. There are 20 proteins which make up close to 92% of the plasma protein composition, and very little, the proteins of our interest may be present in very few amount. So to deplete that or to enrich our protein of interest, a lot of people do depletion strategies where they try to deplete the albumin content, or try to enrich for the proteins of choice by using an antibody-based pull-down methods. So exosomes has a good advantage already here, because when we isolate exosomes, we kind of deplete a lot of these abundant proteins already. So it's naturally a very good source of disease or biomarkers studies. So for this case, we started using plasma. So as we all know, when we did the differential centrifugation, we isolated the exosomes, did a proteomic analysis of these vesicles, and we found out that the exosomes isolated by the differential centrifugation was bit crude, and it had a lot of proteinaceous background, which is coming from albumin and many other plasma abundant proteins, right? And when we did the immunoaffinity pull down, which again did not work as well as it worked in cell lines, probably because of the unavailability of EpCAM positive exosomes, because these plasma are from normal person and is not from patient samples. We pulled plasma from a lot of normal people, and then we started doing these analysis. But OptiPrep density gradient centrifugation was the better method compared to these three, where we could isolate better exosomes, which is devoid of some of the contaminants that you normally see in plasma studies, including albumin and other abundant serum proteins. So the take-home message from this slide is the methodology of choice depends on the sample that you are using, and also the purpose. For example, if you're interested in a functional study, or interested in something characterizing these vesicles for better understanding, then you would better do a better purification analysis like an OptiPrep density gradient centrifugation. Or if you want something like, I want it to be quick and robust, where I have a lot of patient samples, I can't do density gradient, and the sample amount that I have is very low, then you end up choosing other methods like immunoaffinity pull down, or the commercial kits which are available, which are quick and dirty, where you can isolate things faster. But again, we all accept that they are pretty dirty, where you also isolate many other protein contaminants other than extracellular vesicles. So now, in addition to that, this is one question which we always get in many conferences, and even in many other reviews as well. So how stable are exosomes, right? So we talk about exosomes, extracellular vesicles, and we talk about that we can use them as biomarkers. And we all know there's a lot of patient samples which are being stored in repositories. You can collect samples which have been 20 years old or 25 years old in repositories. And can we use these samples? Can we go back to the clinicians and ask all fresh samples, or how long the exosomes are stable? Is there any integrity problem for them? Is there some of the proteins which are kind of getting degraded? So to address this question initially, what we started doing is, when we started isolating exosomes from the plasma, we observed that we did not find some of the canonical so-called markers which were being referred to at that point of a time, like ALIX and TSG101. We could not find ALIX and TSG101 in normal plasma, but in patient plasma you can detect them in a minimal amount, but not in normal plasma. So we started questioning, why are we not seeing ALIX and TSG101? Are we isolating vesicles which are not exosomes, or is there any other type of vesicles? Or is the vesicles bursting out, or they're not stable in plasma? There are a lot of questions to address for this. So we took an approach where we wanted to have a standard, a spike in standard, to see whether these are stable. So we took exosomes from this colorectal cancer cell line LIM1863 and then spiked them into plasma, 1 ml of plasma, and then we stored them at various temperatures in triplicates for varying days. As you can see, we store them in 10 days, 30 days, and 90 days, in 4 degree, -20, -80, and also in 37 degrees Celsius. Then at various time points, we recovered this plasma and then spin it down to isolate it by the differential centrifugation method. And then we probed it for Western blotting for TSG101. Because TSG101 is abundant in the exosomes that we spiked in, and we were able to identify TSG101 in the isolated exosomes which were incubated with the plasma. So when we recovered these exosomes it was clear that we were able to see that exosomes are stable for 3 months, and they were very stable in -80, followed by -20 and 4 degrees to an extent, but not for long in 4 degrees. But -80, they were quite stable for 3 months. So now let's go to the next topic in terms of what is the current study. Most of them, what I presented was published, but I just wanted to give a brief overview. But in this case, we all know there are 3 types of extracellular vesicles based on the biogenesis. We're not talking about the sample type, we're not talking about what cell it is secreting, but based on the biogenesis that we know. One is exosomes, which are small vesicles secreted by the multivesicular bodies. And then there is ectosomes, or shedding microvesicles, which is coming from blubbing from the plasma membrane. And lastly, the apoptotic bodies, which are coming due to apoptosis. So the problem currently in the field is, like, we don't know most of the characteristics of other vesicles. We are well established, or some of the characteristics of exosomes is relatively well understood compared to ectosomes and apoptotic bodies. We know the size is between 30 to 150 nanometer, because when we do the nanoparticle tracking and many other size-based analysis, we now realize that these exosomes can go up to 150 nanometer. And then the shedding microvesicles, this is what the consensus is, that it can go from 100 to 1000 nanometer, and the apoptotic bodies can go up till 5 micrometer. And we know about the biogenesis pathways. The density of exosomes is 1.10 to 1.14, which is considered to be in the field, but we don't know the density in which the ectosomes or the apoptotic bodies settle in. And some of the markers that we think, which we were currently thinking, was ALIX, TSG101, CD63, CD81, and CD9. But ectosomes, we don't know about the markers, and also apoptotic bodies. And if you want to read more about it, you can see Lotwall et al., which talks about the minimal requirements of publishing on extracellular vesicles, where they talk about the problems in terms of identifying markers. So even though we think ALIX and TSG101 are markers, but they are exosome-enriched proteins, and they are not markers. They're also present in ectosomes to an extent. So the take-home message here is EV subtypes are not well characterized. And then despite separate biogenesis pathway, the definitive markers are non-existent. This is one of the burning questions in the field, and a lot of people are trying to address this. So are the markers important, right? When we talk about the extracellular vesicles, the exosomes and ectosomes, do we really need to have markers? Do we need to differentiate them? So again, it depends. Like as I said, how the isolation method depends on the on what you're trying to do. Similarly, the EV subtype is also important in many cases. Like in some cases, let's say we are only looking in terms of disease biomarkers, where we are only trying to identify whether we can find something to discriminate between a normal and a diseased patient. In those cases, I don't think it matters, because as long as you can differentiate between a normal and a patient, regardless of what vesicle type it is, it's still good. But in some cases, or in most of the cases, we can say it does matter, because if we wanted to identify the functionality or what these vesicles play, it's important to purify these vesicles to homogeneity. And then we can study the precise functional role of these EVs and even understand more about the biogenesis pathway if we exactly know the precise markers. So as we said, it is important based on what you're trying to do. So what we did for this particular project is we isolated extracellular vesicles from 3 cell lines. We chose 3 cell lines, which is 2 neuroblastoma cell lines, which is commonly used in many labs. One is the SHSY5Y, and then the SKNB2 cell line, which is the more aggressive neuroblastoma cell line, and then one colorectal cell line called LIM1215. So we isolated EVs by the commonly used methodologies where we used the differential centrifugation. We spun the culture media after 24 hours for 500,000 G for 10 minutes, and then 2,000 G for 20 minutes, just to pellet down the cells and then the apoptotic blebs or bigger the debris which are coming out from the apoptosis. And we discard those pellets, and then we get the supernatant, centrifuge it to 10,000 G to isolate the bigger vesicles, which could be called as the crude ectosomes, the pellet. And then we get the supernatant and centrifuge it to 100,000 G to 1 hour to get a crude exosome pellet. So we also take the supernatant prior to that and centrifuge directly to 100,000 G without doing a 10,000 G, and then subject that to optiprep density gradient centrifugation. Here you have a mixed population of smaller and bigger vesicles, and we wanted to separate these and see what kind of vesicles we get from the optiprep density gradient centrifugation. So one important thing for all these analysis, any kind of extracellular vesicles that you do, is it's important that you do a cell viability assay. So you need to know what is the cell that you normally have and what is the cell that you have at that particular point. So which we routinely perform in the lab. Here you can see the percentage of viability, cell viability in SH5Y, the neuroblastoma cell line, where the cells are surviving more than 91% viable cells. And then, as you can see, we also used, instead of serum, fetal calf serum, we used insulin transferrin selenium, which is another component which can be substituted for, instead of serum, if you want a serum-free media, we use it where it has a lot of growth factors which can stimulate the cells. Again, I emphasize caution in using this because you need to first see some cells don't like ITS because there's more apoptosis in them. Some cells like it, but the insulin also promotes a lot of proliferative signal in there. You need to have a proper control while doing this. While you can see when we compare FCS and also with and without ITS, we can see there's not much cell death. But when we incubated that with doxorubicin, you can see there is cleavage of caspase 3, and there is cell death at 48 hours. You can see by cleave part as well. So again, it would be good for any of you if you're trying to do any assays or any functionalities with extracellular vesicles, you better have to have a control saying that your cell viability is quite good. So then we took the extracellular vesicles, which was spun to 100,000 G, and then we separated them by optiprep density gradient centrifugation. And isolate the fractions and probe for a Western for ALIX and TAG101 in different fractions. As you can see here, ALIX and TAG101 were enriched in the 7th fraction, which is 1.10, but was present in multiple fractions. So the optiprep density gradient centrifugation that we performed was done based on sedimentation, which is meaning you're loading the sample on top of the column and not on the bottom, where you put the sample first and then load all the iodoxonal gradient. And also you can see the 10K or the ectosome pellet, where you can see ALIX and TAG101, the so-called markers of exosomes, are also present in this ectosomal population or the crude ectosomes here. So in the lab, as we all know, we struggle to isolate exosomes. When I say struggle to isolate exosomes, we do a lot of cell culture, we have a lot of plates, we culture a lot of cells to get enough amount of exosomes to do the whole prep. So we can't afford to do or spend a lot of protein in multiple methods like BCA or Bradford. So what we try to do is quant the amount of protein that is present in extracellular vesicles by sensitive methods like CypherRuby. So this methodology helps us to quant in picograms level, where we use very little microliter of sample, and we can quant the amount of protein relative to each other. So this is an image that you're seeing on where we quantify the amount of protein that is present in the fractions that was collected by from OptiPrep density gradient centrifugation. As you can see, the seventh fraction, which had the Alex and TH101, where had high amounts of protein in one of the runs, and then we repeated this multiple times based on we optimized the density gradient, where we are working with a different gradient centrifugation system. Instead of 5 to 40%, we are playing up with that density gradient, and we are able to get a high amount of protein in the 10th fraction, which is the higher density fraction. As you can see, the exosomal fraction is 1.10, which is enriched in there, but there is also a lot of protein in 1.12, but there was less Alex and THG101. So we repeated this multiple times in many other cell lines and even in 5Y. As you can see, that the 10th fraction always had more amount of protein, and which is corresponding to 1.20, but the exosomal fraction had some protein, but mainly the sorry, one second, it's a motion detector. Sorry, apologies for that. So it has a motion detector, so it didn't detect anything. So as you can see, the 10th fraction had a lot of protein in there, but we are curious to see what are the vesicles which are present in the 10th fraction. So now you can see that when I go to the Western, you can see that the Alex and TH101 are enriched in the 7th fraction, but the 10th and the 9th fraction had a very low amount of Alex and TH101, even though they had a lot of protein. Right? So they had a lot of protein, but then what are these proteins? So we were curious to see, and then we wanted to characterize these EV subtypes. So we took those fractions, 1.10, which corresponds to exosomes, 1.14 and above, which corresponds to the bigger vesicles, and then we also had a crude 10K pellet, and then a whole cell lysate as well. You can see that the Alex was present in both, but the marker, which is the GM130, which is supposed to show that if there is any apoptosis in there, it was not present in any of those things, suggesting that the isolation was pretty good. So then we went on to characterize these extracellular vesicle subtypes by biophysical methods. So the biophysical methods are important to show the size and shape of these vesicles. As you can see here, the 7th fraction, or the 1.10 fraction, which was isolated by the OptiPrep density gradient, you can see the vesicles which are smaller. But when you went to the higher density fraction, you had bigger vesicles which were more than 200 nanometer in diameter. It was consistent also in the 10K pellet, or which was like the crude ectosome pellet, which you call as such, where you can see the 5Y cells had bigger vesicles, and then even the LIM115, the colorectal cancer cell line isolate, also had bigger vesicles. What was interesting, you can see that is these vesicles are not as homogeneous, and you can see there are bigger vesicles where they are stressed because of the centrifugation protocol. And when we see that in the OptiPrep density gradient centrifugation, you can clearly see that some of the bigger vesicles don't make it up 100,000 G. They kind of burst, which we know, and they don't sustain those pressure when they are going to high-speed centrifugation, and they burst up. So it's important to understand, like, these bigger vesicles, the crude, even though it isolates a lot of them, there's also some contamination in there. So apart from EM, we also wanted to characterize these from atomic force microscopy, which gives a nice overview of the vesicle size. As you can see, we took the fraction 7, which is also the exosomal fraction, and we quantified what is the size of these. And as you can see, most of the vesicles which was present in the smaller density fraction or the lower density fraction, these were similar to exosomes, which were ranging from 40 to 120, 130 nanometers in size. So when we took the higher density fraction and then did the same analysis, you can see that these were again consistent with the electron microscopy analysis. There were a lot of bigger vesicles which were more than 200 nanometer and 500 nanometer in diameter, which consistently said that these are two different types of vesicles. So we did characterization in terms from the atomic force microscopy, where we wanted to quantify the number of vesicles which were in the size range. As you can see here, 98% of the vesicles which are present in the lower density fraction were in the range of 30 to 150 nanometer in diameter. However, in the larger density fraction, most of the vesicles were more than 150 nanometer. You can see close to 90% of them are more than 150 nanometer. Only 10% of the vesicles were less than 150 nanometer. So when you see this analysis, you can see that there is still some vesicles which are smaller than 150 in the higher density fraction, and there is also some vesicles which are bigger than the 150 in the exosome-like fraction. So I can conclude from this analysis that we have not isolated the vesicles to homogeneity, but we have enriched for them. So that's the take-home message from this. But we wanted to identify what could be used as markers. Can we find some markers of these ectosomes which were not present in exosomes, and vice versa? So we started calling these small EVs or exosomes and large EVs or ectosomes based on the size range. And then we subjected these to proteomic analysis. So now we wanted to see what are the proteins that are present in these vesicle subtypes. So then we can make a clear idea of what proteins are present and what can be called as markers. And as we have done this on 3 cell lines, where we isolated exosomes, ectosomes, and 10K, we can now see it is general to the cell type, and we can come up with a list of markers. So as you can see in this slide, compared to the whole cell lysate, there are 591 proteins which were enriched or highly abundant in exosomes. Similarly, compared to the whole cell lysate, there are 558 proteins which are highly enriched in ectosomes. This clearly says that there is some kind of sorting or selectivity mechanisms which is happening in sorting the protein cargo into these extracellular vesicles. As a separate project, we are also very much interested in identifying what are the sorting signals that kind of targets or directs these proteins into the exosomes and ectosomes. Most importantly, when you compare between exosomes and ectosomes, you can see these are a clear, distinct proteomic profile, where you can see that 450 proteins were uniquely present in exosomes, while 612 proteins were uniquely present in ectosomes. This clearly says that we can come up with a list of markers, but it's important that it follows up in multiple cell types rather than one cell type. That's why we wanted to do more than 2 and 3 cell lines. So now, how do we make sense of this proteomic data? So now we took those proteins, and we know that the exosomes are indeed formed by the escort machinery or the multivesicular bodies. So we looked at the proteins which are involved in the escort machinery and plotted them as this histogram, where you can see in terms of exosomes, the blue color represents the fold ratio of a protein in exosomes compared to ectosomes, meaning here the allix is present more than 10-fold, it's 10-fold abundant in exosomes compared to ectosomes. Similarly, exosomes compared to the 10K or the crude exosomes. You can see in this figure that allix is more than 4-fold highly abundant in exosomes compared to the 10K. So likewise, when you plot it like this, you can see many of the escort machinery proteins, including TSG101, the VPS class of proteins, were mostly enriched in exosomes, which kind of kind of says that the escort machinery is indeed only involved or as much involved in exosome biogenesis and not as much in the ectosomes. And then the proteins which are exclusively identified in exosomes are denoted by an asterisk here, and then a hash is given those proteins which are only detected in ectosomes or 10K. There is not much for that. Only VPS37D was identified only in ectosomes and ectosomes, but as you can see, it does not qualify the twofold ratio, so we did not consider that to be significant. But those which classified more than twofold, we considered that as significant. You can see VPS32B or CHIMP4B was only present in exosomes, not identified in exosomes or ectosomes at all. We further went on to do the tetraspanins. We can know that we do know that the tetraspanins are important in exosome biogenesis, and they're also being proposed as exosomal markers. So we looked at the profile of them, and these are membrane proteins, the tetraspanins, which are 4 membrane spanning domains. But when you look at the exosomes and ectosomes, the ectosomes are bigger vesicles while the exosomes are smaller. So you tend to see the surface area for exosomes and exosomes. There's difference between them. So you will identify more membrane proteins in exosomes compared to ectosomes. This goes in saying, because if there is a surface area which is kind of you can enrich for the membrane proteins in the exosomes because they have very less surface area compared to the ectosomes, while we get a lot of cytosolic and other intraluminal proteins in the ectosomes, and this also goes in saying for the whole cell lysate, when you tend to identify more intracellular proteins as opposed to the membrane proteins. So this could be one bias in the study, but having said that, we can see that the CD81 protein was mainly identified in exosomes or exclusively identified in exosomes, but not in the ectosomes. But the other proteins, CD63, CD9, and other tetraspanins were there, but the most significant one for ours is CD81, tetraspanin 14, and tetraspanin 9. But again, when you understand how mass spec works, again, mass spectrometry kind of technologies, even though they can identify a lot of proteins in no time, but they also have some inherent problems where they suffer in terms of identifying some of the hydrophobic amino acids easily. So for that methodologies, we have to enrich for membrane proteins, but that could be one of the bias, because why we did not identify CD63 and CD9 in the preparation as much as compared to a Western. Again, grab GTPases, which is one of the class of molecules which are involved in exosomes and ectosome biogenesis. You can clearly see here that there is a set of molecules which was exclusively present in ectosomes, this set, the RAB molecules, and those RAB molecules which were only exclusively found in exosomes. So there is a set of molecules which are performed exclusively in there, and this was consistent in 2 of the cell types. Most of the proteins, not all of the proteins, most of the proteins were consistent where it was only present in one of the extracellular vesicle cell type. We're currently doing some knockdown analysis and knockout analysis in cell lines to identify what is the function of these RABs in exosomes and what are the function of this subset of RABs in ectosomes. And there are other proteins also which we know about, annexins and flotulins and the integrin betas and the RAMP molecules. We also profile that and see what is the profile in terms of exosomes and ectosomes. Integrin beta was only present in exosomes, but not as much. So based on our current knowledge from all these slides, it's very clear that what we know is mostly or biased towards what we know is biased towards exosomes. We know a lot about exosomes compared to ectosomes. As you can clearly see that all the proteins that we know of were more enriched in exosomes, and we completely don't know anything about ectosomes. So now we wanted to see the other side of the picture. So what about ectosomes? As I said, we don't know too much, but there are a handful of studies, like 5 to 6 studies, which have done on ectosomes, and we wanted to do literature survey. We went on to collect all those studies. We curated those data. When I say curated the data, we looked at the Excel sheet and then we made up the proteins which were known to be identified in ectosomes or involved in the biogenesis of them. So one of them is ARF6, as we know of the papers, and flotulin-1, integrins, MMP2 extracellular protease, and VAM3. So these are the list of proteins that we came across, and for the entire list, you can download in this particular oncotarget paper, which is currently in press. So as you can see, these are the only handful amount of proteins that we could identify, but all of these proteins were more enriched in exosomes compared to ectosomes. Right? So which is even more shocking, because some of these proteins we thought are ectosomal proteins, but they're not. But MMP2 is the only protein which was exclusively identified in ectosomes, but not in exosomes. Right? This is extracellular protease. It can be secreted by the classical secretory pathway, and it could be an associated molecule also in terms of ectosomes. Why it is associated only in ectosomes and not in exosomes is a different question altogether, but we are trying to address that as well. So now, based on the proteomic study, this is what we kind of think. These could be some of the potential markers of ectosomes. I name potential because some of these markers may not stay same when you test multiple cell types, but CD81 was consistently present in exosomes but not identified in the larger vesicles. Similarly, we got the VPS, the escort machinery, EPS 24, 36, the tetraspanins 9 and 14, the syntonin and the integrin alpha 3. For ectosomes, we think MMP2 is one of the good molecule, but similarly we have the RAD GTPases, and then the spectrins, the mucins, and cytokeratin 18, and the ribosomal proteins 9 and 18. So there's also two other studies which are performed by colleagues. One is from Clotilde Thierry's group. Most of you know her. She is a well-known scientist in this field who has been working on this field for a very long time. And they have also isolated exosomes and larger vesicles, and their results also correlate exactly with in terms of CD81, and they have done a nice study also where they have isolated exosomes from multiple cell types, and they have proven that these markers could be validated in Western. But you can study her paper when it comes out in more detail. Currently, probably it's in review or in press. Similarly, another group from US, Dolores Divicio from LA, she also isolated exosomes and oncosomes, and she was able to show that cytokeratin 18 was present in the larger vesicles and was absent in exosomes. But one thing which we noticed, this keratin 18 is an epithelial marker. So fibroblast cells may not secrete them, so it cannot be used as an ectosomal marker in a lot of cell types, but at least in epithelial cells, we don't see keratin 18 secreting in multiple exosomes, but mainly in ectosomes and epithelial cells. You can see a Western here to confirm that. I can see the GM130 was not found in any of the vesicle fractions, but only present in whole cell lysate. But the MMP2 was found only in ectosomes and 10K, but not in exosomes. Similarly, the CD81 was only found in exosomes and not in ectosomes and 10K, again validating the proteomic analysis that we have done. So all of this data for this 3 cell lines is available as supplementary information in the OncoTarget publications that I presented. So we've moved on further to study what is the functional role of exosomes and ectosomes. Because we know about something about the proteomic profile of exosomes, but what do they functionally do? Is there any functional distinction between these two vesicle subtypes? So we used this program called FundRich, which was developed by a lab, which does the functional enrichment analysis, where you can put your extracellular vesicle data from whatever omics technologies you do. And then you can do an enrichment analysis of what are the classes which are underrepresented or overrepresented in your class. And it can give a p-value and a nice figure, a publishable quality, where you can put that in a manuscript, and this is what we have done. As you can see, the escort machinery was enriched in exosomes and not in the ectosomes. And similarly, many of the signaling molecules, the plasma membrane proteins, all of them were enriched in exosomes compared to the ectosomes. In ectosomes, we get proteins implicated in gene expression, translation. Those were the molecules which were enriched in them. Again, as I said before, the enrichment of the plasma membrane proteins and signaling molecules could be partly due to the surface area of exosomes and ectosomes. That could be one reason. But we also think that exosomes probably have more signaling implications than ectosomes from the proteomic profile that we see. Then we also looked at the biological processes that are enriched and depleted in exosomes. Here we are comparing exosomes versus ectosomes. You can see the metabolism, proteins which are implicated in metabolism are depleted in exosomes while they are enriched in ectosomes. But as I said, proteins which are implicated in cell proliferation, signaling, immune response, signal transduction, protein transport, and enzyme receptor signaling pathway were all enriched in exosomes compared to the ectosomes. And now this is another slide which shows the molecular function, which are the molecular function terms which are enriched and depleted. Similar to that, you can see a lot of enzyme activities are enriched in the ectosomes. But here again, the transmembrane protein, the signaling axis is kind of enriched in the exosomal cargo. So now we have done this one, but we wanted to identify the mutant proteins which are secreted via EVs. So again, when we talk about exosomes and ectosomes, we all know there are a lot of disease proteins which are implicated in disease being secreted out of EVs. So we have to have a prior knowledge. For example, if you're looking at a protein EGFR mutation, we need to have a prior knowledge for looking into what mutant protein or what oncogenic molecule is secreted out of EVs. But is there a methodology where we can identify these in an unbiased, in a global way? For that, we started using the proteogenomic analysis, where we correlate the proteomic data with the genomic and transcriptomic data to identify the mutations as well as to enhance the genome annotation qualities. So this particular methodology has been available for quite some time in the field, but nobody has used this in the context of extracellular vesicles. So we said, I published a paper in 2012 where I used this particular methodology in identifying fusion genes, mutated or truncated proteins in colorectal cancer proteomic samples. I published that as a methodology, and those interested can go and look at this particular article. But I didn't call it as proteogenomics at that time, but we call it as identification of mutant proteins at that time. But now the word proteogenomics have caught up, and then people are using it. So the take-home message is we used the proteomic data from 18 colorectal cancer cell lines, identified the mutant proteins which are being secreted out, and we also described the method of how we did that. So similarly, what we did in this case is we had the neuroblastoma cell line SH5Y, right? And we did an exome sequencing of that particular cell line. So what exome sequencing does is it will identify all the mutations which are present in the coding regions or what you think is the gene regions which codes for a protein in the particular cell line. So once you have the coding mutations, which could be the single nucleotide variations, or it could be the insertion and deletions in the particular genome, we made a protein database of those of the particular exome sequencing data. Then we isolated exosomes, ectosomes, and then the whole cell lysate, did a proteomic analysis as I showed before, and we searched this proteomic data with the mutant database, and we ended up identifying a lot of mutant proteins which are secreted out of exosomes and ectosomes. So this is a powerful technique. Like in terms, if you want to identify which could be a mutant protein, we can use this easily because now the exome sequencing data is available for a lot of cell lines publicly. And then all you need to do is have a proteomic data from your exosomes or your ectosomes, and anybody can do this analysis, and then you can identify the mutant proteins that are secreted out of these vesicle types. So this is a representation, a circus plot of the mutations that we have identified in this. Again, this is a pretty picture, but you can zoom in and look at it. But in this case, you can do this kind of representation is what I'm trying to address by this particular slide. So what we identified is through exosomes, we saw a lot of proteins, which are mutant proteins, which are secreted out. All these proteins have been implicated in oncogenesis, drug resistance, and a kind of metastasis as well. Similarly, we also identify a set of proteins which are secreted out of ectosomes. We are doing functional analysis of what is the function of these mutant proteins in the recipient cell. For example, one of our interests is Wnt signaling pathway, and we have looked and identified that the mutant beta-catenin is secreted out from colorectal cancer cell lines. And when we take these exosomes which contain the mutant beta-catenin and incubate that with the recipient cell, we can induce Wnt signaling pathway in the recipient cell. So this is beautiful. When you say there's a mutant protein which is constitutively active, right, and that can be transferred to the recipient cell, and the function of that particular protein is again amplified in the recipient cell. And imagine that in the tumor microenvironment, and that is immense, where the cancer cells can evade multiple mechanisms where they can keep the cells in terms of the proliferative mechanism for a constant time. So again, the proteogenomic analysis of EVs, it can be used to identify mutant proteins, and you can identify disease-specific biomarkers. Like say, if you have a mutant protein, the lot of biomarker field which we talk about, we always have issues with specificity. How specific is a biomarker for a particular disease, right? So now when you identify a mutant protein or a mutant RNA, which is being transferred via the EVs, and it's also identified in plasma or many other bodily fluids, you would get the specificity which is currently lacking in many other kind of techniques, because these disease-specific or the mutant protein or RNA would be made up only in the disease cell, right? So you would get the specificity that is required. So you can read these two reviews if you are interested in this particular analysis. So now we wanted to see what is the oncogenic potential of these EVs. So we, as I said before, the exosomes have a lot of signaling proteins in them, but the ectosomes did not have much. But when we looked at the cosmic database, which has a lot of oncogenic mutations which are present in many cancer types, we downloaded the data. We only took at look at the significant list and overlaid those proteins which are known to be implicated in any particular cancer. For example, this is the list of proteins which are implicated in thyroid cancer, and we overlaid that on exosomes and ectosome data from our proteomic results. From this, you can see a lot of blue compared to red. The blue means this is exosomes, and the red is ectosomes. We can see most of the proteins which are implicated in cancer are all identified or mostly identified in exosomes compared to ectosomes. And this clearly says in this particular thing that exosomes have more oncogenic cargo than ectosomes. We further went on to do the same analysis with EST data, which is expressed sequence tags, and that is also analyzed from normal tissues as well as from cancer and disease tissues. As you can see consistently, these proteins again are more found in exosomes compared to ectosomes. So these are bioinformatic analysis, and the statistical value was quite good indeed, and it convinced us enough to do some experiments and to see what is the difference in terms of the oncogenic potential between these two vesicle types. So we did a proliferation assay where we isolated exosomes from the 5Y cell lines. We incubated that with a neuroblastoma cell line called BE2, which is already an aggressive cell type. As you can see here, the untreated cell is here, it's the control. But when you treated the cells with ectosomes, the fold proliferation went up. But the exosome treatment, you can see there's more than twofold proliferation, and it was a significant one compared to that untreated cell. When you compare between exosomes and ectosomes, that's definitely not a big significant difference between these two, but exosomes were significantly inducing more proliferation in the recipient cell compared to the untreated cell. The biggest question on these kind of assays, right? So because we incubated with 40 microgram per mil, but having said that, is this concentration physiologically relevant, right? So we kind of incubate 200 microgram, 300 microgram, 400 microgram at any one time point, and then we say, oh, we see all these functionalities. But I think we should be very cautious. We should be very cautionate about what we are trying to do, because some of the concentration that we are using may not be physiologically relevant. Because the biggest question currently in this field is what is the concentration of the exosomes or the EVs that is being secreted out in the cells in vivo, and what is the physiological concentration? So for until we answer that, it's better to go with as lower as we can. And we also can use higher concentration, but try to use a lower concentration also and show that the signal is starting from there, and not to try to use a higher concentration and stick with the signal. So we also went on to do a migratory analysis where we wanted to see the role of exosomes in inducing migration. As you can see here, the untreated cells is used as a control, where you can see there was a scratch in the cells, and then we left it for 24 hours and then saw how the wound was healing. And when we treated these cells with ectosomes, you can see the wound was closing, but not as significantly. But when we treated the same with exosomes, we can see there's a significant difference. And you can see the wound was even closing more, clearly showing that the exosomes induce more migration than ectosomes. We also did that with 10K, the crude exosomes, and we repeated this with, as I said, here you can see it's 40 microgram. We went to a lower concentration of 20 microgram per mil, and then you can see here the 10K and the ectosomes did not have a significant effect, while the exosomes indeed had a significant effect in inducing migration in the recipient cells. So this confirmed that, consistent with the bioinformatic analysis, that the exosomes are more oncogenic, where they can induce more migration and proliferation. While proliferation cannot be tagged as an oncogenic potential, but the migration is. We also did an invasion assay where we also showed that exosomes could have more invasive potential than the ectosomes. But the summary, to give a summary of the talk, so you can see ALEX and TH101 are enriched in exosomes and not markers of exosomes. So you can call them as enriched proteins, and you can see the minimal requirements by Lotwall and Clotilde and by the scientific board of ICEV, where they talk about these particular thing that ALEX and TH101 are enriched. CD81 could be used as exosomal markers, and MMP2 as ectosomal markers. But again, more studies need to be done. As of now, with 3 studies, we can see that CD81 is quite distinct. But MMP2, we have done it, but in 3 cell types, but we need more studies on this as well. And exosomes are enriched in membrane proteins and signal transduction molecules compared to the ectosomes, and exosomes are more oncogenic than ectosomes. So with this, I'd just like to acknowledge the people who did the work. The 2 postdocs, Shivakumar, Kirtikumar, who is a bioinformatics postdoc, and Lagiru, who did all the experiments involved in this project, along with the students Michael Lim and Emily. And thanks the funding agencies, and thank you all for listening, and happy to take any questions.
Very good. Thank you for that highly informative presentation, Dr. Mathivanan. We've already had more than 60 questions come into our feed here so far, so a very stimulating presentation, to say the least. For those of you who have not had the opportunity to ask a question, please use the blue Q&A button at the lower left of your presentation window. We'll go ahead and get started on the Q&A session. The first question is, how does the isolation of ectosomes and exosomes affect the vesicles' function? Can one expect to isolate and then reintroduce them into cell cultures or a model organism and get physiologically reliable results?
Yeah, thanks for the question, Chad. Thanks for the question, Chad. The question here is, how does the isolation of exosomes and exosomes affect the vesicle function? Can one expect to isolate and then reintroduce them into the cell cultures or a model organism and get physiologically reliable results? And that's a good question indeed. The exosome isolation method definitely will affect the function of the vesicles, because now when I say the function of vesicles, when you choose a method of isolation, it depends on what vesicle you're getting. For example, if you're doing an immunoaffinity method, right? So for example, you're isolating exosomes by CD63 positive exosomes, you're only going to isolate CD63 positive exosomes, and then you're neglecting a population of CD negative, CD63 negative exosomes. So when you take the CD63 exosomes and then you incubate on the target cells and look for a physiological function or any function in that setting, you're just going to bias the whole thing, because your methodology that you used was an immunoaffinity and you ignore the other population. Similarly, when you on ultracentrifugation, again, you might pellet down some of the complexes in there, so you might bias the results in a way. So the answer to the first part of the question is yes. If you will, the methodology that you're using is very important for the vesicle function. And for the second question, in terms of do we get physiologically reliable results when you're incubating on the cell cultures and model organism? Again, this is a question of which everyone is trying to understand, because we don't know the physiological concentration. One thing we know is exosomes or any other EVs that we think is constitutively secreted. But now we are giving, when we're doing all this assays in vitro, we are incubating the exosomes with the target cells for one time point, and then we are giving one dosage. Right? So sometimes we give up more, sometimes we give less. As you can think, this is happening constitutively in vivo. You can think that can compensate to an extent. But having said that, that question is debatable, and we don't know the answer for it yet. So that's one question which a lot of people are trying to answer, and I'm thinking that's one which is not going to be easy to answer, because for first we need to understand what is the physiological concentration of the EVs in the normal, and then what is the role of the EVs in physiological conditions, even in homeostasis and things like that, before trying to go there. But very good question indeed. We need to keep that in mind before performing or designing any assay. Thanks.
Great. Thank you for that response there. Several questions have come in regarding the stability of exosomes. Stability in cell media, in your freezer, and freeze-thaw cycles as well. Can you comment on those?
Yes, so that is one important question as well. So prior to that, so the stability, there are 3 or 4 papers which are currently out there in the literature where it talks about stability. People have done the freeze-thaw cycle, where they kind of take exosomes from the freezer and do a repetitive freeze-thaw cycle, but does not affect the exosome stability as much. People have also incubated the exosomes in -80 for a longer period of time and looked in terms of the protein markers inside them, we are one of them, and we can say that it was stable. However, we haven't looked at the membrane proteins. So the membrane proteins, we don't know how intact they are and how well they affect the uptake. But when we did an uptake assay for those exosomes which were preserved or frozen down for 3 months in -80, they were having similar uptake efficiency as that of the cell, that of the other exosomes which was given on the 0 day as well. As I said before, the functionality, right? The functionality is a different question altogether, where a lot of people have shown that the exosomes are not as functional as they used to be when they are at 0 days, when you freeze them for a longer period. So we see that consistently as well. When we keep them for up to 3 to 4 weeks, we are getting some of the functionality, but when we do more than that, we don't see the same amount of response which we used to see before. So what we do currently in the lab is we try to not keep the exosomes if it is a functional study for more than 2 to 3 weeks, maximum 2 weeks. We try to keep that. But if it is a biomarker or a proteomic profiling of those kind of study, then again, you can probably go up to 3 to 4 weeks. But it's advisable to do these kind of assays as and then. But the good thing is the exosomes are much more stable than what we thought of. They are really stable. But having said that, we need to have our precautions as well. Thanks.
Great, thanks for that. We are running low on time. We'll ask one more question here, and that question is, does the proteome analysis of exosomes give any clues about the cell entry mechanism?
Yes, that's a good question indeed. That is, again, one of the active areas that my lab is concentrating on. With what does, because now when you see at the, when you see the proteomic profile of EVs or exosomes in general, you change the growth factor, you kind of change any kind of setting, you induce hypoxia, you kind of give, you knock out some protein. The proteomic profile completely changes irrespective of what this is. So it is clear that the membrane proteins are also changing. As we all know, the membrane proteins have a more important role in the target cell fusion, uptake, or in terms of entry. That can give us clues. But having said that, there's also a lot of proteins which are known to be involved in biogenesis. But as the question is in terms of clues, yes, it does give clues. But having said that, you need to be careful, because now you have a lot of proteins which are generally present in exosomes which can have biogenesis, may not have an effect in terms of uptake. Plus also there are some proteins which are specifically present for a particular cell type. So we kind of do knockout analysis of some of these membrane proteins and see how the uptake specificity changes. Yeah, that's pretty much where it is. We definitely have clues from the proteomic analysis and even from the lipidomic analysis, how the lipidomic profile changes, and then the uptake efficiency changes as well. Thanks.
Great, thanks again. Unfortunately, we are out of time. I want to thank the audience for their interesting questions and participation in today's event. I would also like to thank our sponsor, Beckman Coulter, and of course, Dr. Suresh Mathivanan for making today's educational webcast possible, which will also be available for on-demand viewing through beckmancoulter.com. Thanks again. Goodbye.